What is this?
¿Esta termita qué es?
This may be the big beetle that we're looking for here. Either it's a Phleoborus or it's a Loch Ness monster. One of those two. Look at that. So we have a male and a female of Phleoborus, the biggest bark beetles in the world. The female makes this hole, but it's not very long. It's not like a typical gallery. And there she deposits her fungus, which it's oozing from her mycangia. It's visible by naked eye. So this is the beetle. If you zoom in, this is the mycangium, the fungus pocket, and you can see the fungus, the white thing, growing literally in it and out of it. This beetle is walking with the fungus garden in its body. And the fungus infests the wood around and does something to it. We have no idea what it is. We don't even know the fungus. And the larvae then eat it, which is interesting because like what fungus turns wood into an animal diet?
Yeah.
I mean, I'm thinking like we should, you know, I don't know, turn that into human food or something. This is crazy, this science fantasy. So we're at the University of Florida Forest Entomology Lab, right? And we're not in Florida right now, we're in Guatemala in a super cool remote station called Yaloch. And the main goal of our endeavors here is to find dead trees colonized by beetles that are called Platypodinae beetles or ambrosia beetle pinhole borers. They're actually the oldest farmers on the planet. They have recruited this fungus, or perhaps the fungus recruited them for transport 80 million years ago. And they are really incredibly diverse in the tropics. So we're studying the association between these insects and the fungi that they carry around to infuse into the wood so that the fungi feed their babies. The whole ambrosia symbiosis, right? Oh, nice. Hey, folks, you gotta see this. This is a photo opportunity first. This is amazing. This is what we want to see. Look at the noodles. Here's the hole. Here's the fuzzy sawdust. You know why it's fuzzy? Because they pull out really long splinters. If you look at the individual splinter, look how long they are. Must be crazy work. So this is a skinny one, probably a Xyloborini, ambrosia beetles. And this is a little fatter, and I have no idea what it is, but just by virtue of being different, it may be worth sampling.
Too, it seems like those
Yes,
were
exactly.
thicker.
Yes.
So when you're doing this, you're kind of cutting on one side, cutting on the other, and basically just like cutting out.
Exactly.
What
So
you're saying is
what
the
we
cookie.
want is to cut out a cookie. Many different tools, hardware primarily. We start with a chainsaw. It's really cool to be sampling these beetles with spider monkeys above our head, and they're not running away. Once you use the chainsaw to make the cookies, you could then use littler tools to peel the wood away and collect the beetles.
As someone that's never really used a hammer and chisel before, I have to be really careful to make sure I don't smash my fingers. Line it up, try to get it right on that crack. Oh, I always feel bad when the wood block gets flung off to the side. I can imagine the beetle, if there's beetles in there, they're probably not having a great time. The thing I really like about this is I feel like an archaeologist finding these civilizations just beneath the wood, and you don't know what you're gonna find. Okay, I got it started. I'm gonna finish it with these clippers. Yes, there is someone in there. It's a female, that's awesome, because the female is the one that carries the fungus that we're after. So you can see all of this work that's gone in from taking a cookie this size down into smaller pieces, but to be able to get one with the female is awesome.
Oh, and here you see a cool thing too. This is a Platypodinae larva. From a tiny little egg into this big chunky protein pouch, they just graze on the fungus. It's a very, very old mutualistic symbiosis, and there are lots of species, 1,200 at least. But they're often, most of them are really specific, really dependent on nice old growth forests. There are not many forests like that left anymore. I know everybody has heard it many times, but I still find it fascinating how in a nice forest, all the biomass is up there and you can walk through it. But if a forest is disturbed, if a forest is jungle at the ground level, it means that it doesn't have enough in the canopy, and it's a secondary thing. But if it's nicely walkable, friendly to people, it's the primary rainforest.
Spider monkey throws poop at me and misses and hits you in the face.
This trip is really special too because I've been to Guatemala before when I was 18. My mom's side of the family is from Guatemala City. And so to come back here to this beautiful country, to explore the jungle, it's been incredible. And I'm loving every second of it.
Sometimes you find like treasures. This is incredible. Look at this. Ambrosia beetles drilling into this tree, and look at the color. Look at the consistency. Bright pink. And the cool thing is that this is how you tell ambrosia beetles. They don't want the wood, they just push it out. They want the space inside so that the fungus can grow through the wood.
It's like someone spray painted it.
Yeah. We are really digging beetles from very precious woods here. The kind of stuff that fancy furniture is made of. You know, the concept of the monster cookie, this is the opposite. This is the, oh, no, the other way. Do you know the concept of the cookie monster? This is the monster cookie. These are the woods that we get to sample in this rainforest. They're called the precious woods, precious timbers. Look how hard it is. It's incredible, like you see us struggling with this. And look, these beetles, so tiny. They go this deep into the wood and make these extensive galleries. How? How do they do this? In a wood that's hard as stone, so it's really hard to even like split it with a chisel. You know why? We know that. Because if you do an X-ray of their head, the ends of their mandibles light up like crazy. They're white because they have metal in the mandibles. So they're super sharp. Literally, they're like drill bits, these beetles. There is so much discovery that one can make, you know, and we're at the very beginning, opening the window into this universe. You know, we used to come to a place like this and try to culture the fungus directly from the insect. It's really hard to turn this into a microbiological lab. And it's not really necessary because we figured out how to make this medium really from sawdust and agar. And our amazing lab manager, Daniel, made a whole batch of them. They look very tasty. And that's a perfect way to transport them internationally with the proper permits and everything. The key here is that the beetles stay happy and alive and they're the best vehicle for the fungus. But they don't know when you just put them in, they don't know what to do. They're like flopping around, ah! So we figured out that you have to make a little dip, like a divot, like a hole with a forceps, clean forceps. Then you take that female and stick her head straight into that artificial medium. And it clicks like that in her little head, and she starts digging immediately. If you don't make that little hole, that little adjustment in the protocol, she keeps flopping and eventually she might die. And then you add the male, and the male starts tapping the female on her abdomen, on her butt really, and communicating, that's me, your guy, let me in. And she does. They work really well. They are doing one thing at a time, but they do it really well. Thank you, people. I think this is it for tonight.
What number was that?
20.
I think there's tiny platypus.
Where? Yes, Toby, you're right. Like half of this is bark beetles. Whoa!
Oh, I see that.
There are so many. How are we gonna ever make any sense out of this?
So now we're back in Florida at the Forest Entomology Lab, and this is where we can isolate the fungi from these beetles once we get them out of the wood tubes. So the process starts here. We take a Dremel and we cut a window, avoiding the beetle because we don't want to kill it. And then we get a ID on what the beetle is. And what we want to culture is the female. She's the one that's carrying that pure culture that she's feeding her offspring. So we'll take the head and the pronotum, separate it from the rest of the body. We grind it up, and then we serial dilute down, and that's what's going to get plated in the hood so that we can start to see the different fungi that are growing and hopefully isolate pure colonies to then sequence to get their identity. And once I've done that, we end up with lots of different fungi on their own plates. These really beautiful, colorful looking fungi are yeasts, but these are not our target fungi that we're after. We're more interested in these fuzzy filamentous ones because we think that these are the primary symbiont of the beetle. This is going to be what the beetle is feeding her offspring.
These look like the ugliest fungi, but they often are the right thing.
Right.
They look so like
Nutritious.
wholesome, exactly.
Yeah. There's going to be like one primary symbiont per like beetle, but to think that like all of these could be co-occurring with each other, and
That's
like what are they doing in there?
We may be looking at some jungle of promiscuity where like no matter what beetles penetrate the tree, the fungi just travel between them. And then it's like this real community that probably keeps coevolving for millions of years.
Yeah.
But we don't know. Isn't that a little scary that, you know, like you go somewhere, you do this work, and then instead of like publishing in paper and answering a question, you open like this window and there is this vast space. Like you don't know what there's this jungle. It's just opening a window to another universe. Every single thing here is like 10 questions on a petri dish. So many questions.